Hello everyone. Today we will discussing the important topic that is inborn errors related with phenylalanine and tyrosine metabolism. Check my voice first. Whether I am audible and visible to you. Just check it. Okay. The topic of discussion is inborn errors of phenylalanine and tyrosine metabolism. These two amino acids considered together because phenylalanine is converted into tyrosine and it gives they gives important compounds. For example, they gives melanin in skin, hair and eyes. They give dopamine, adrenaline, noradrenaline. They give thyroid hormone T3 and T4, and in their metabolic pathway these are both glucogenic and ketogenic amino acid, both glucogenic and ketogenic amino acid. So today we will discuss about the important inborn errors that includes the first and important is phenylketonuria. Phenylketonuria. The classical characteristic of phenylketonuria is mousy odor of urine. Mousy odor of urine and hypopigmentation is there along with the mental retardation. So mental retardation, mousy odor and hypopigmentation. These are the features of phenylketonuria, mousy odor of urine, because of excretion of phenyl acetate in the urine, because of excretion of phenyl acetate in the urine. So this is important disorder, inborn error related with phenylalanine and tyrosine metabolism. It is involved, it is due to the blockade of enzyme which converts phenylalanine into tyrosine. The another name of tyrosine is para-hydroxyphenylalanine. Para-hydroxyphenylalanine. Then the second important disorder, the important disorder which includes 𝐾𝐵. This is again important disorder, 𝐾𝐵. The classical feature of 𝐾𝐵 is black urine. On exposure to air, it turns into black color. Okay, and the patient live normally, asymptomatic at up to the age of 30 to 35, 40 years, and during this period he or she develops arthritis. Okay, so black urine on exposure to air, this is a classical feature of 𝐾𝐵. The defective enzyme is homogentisate oxidase, that we will discuss why there is arthritis, why there is black color urine, how the homogentisic acid is converted into benzoquinone acetate, and polymerization they will give black pigment, that is 𝐾𝐵. So 𝐾𝐵. Then third important disorder related with this pathway is tyrosinemia. Okay, the tyrosinemia, it includes first tyrosinemia type 2. Tyrosinemia type 2. So due to the defect in the tyrosine transaminase, there is tyrosinemia type 2. It is also known as Rischner-Hanhart syndrome, and there is oculocutaneous manifestation, tyrosinemia type 2. Then neonatal tyrosinemia. Neonatal tyrosinemia. The next one is tyrosinemia type 1. The typical feature of tyrosinemia type 1 is type 1. There is cabbage-like odor, boiled cabbage-like odor in tyrosinemia type 1 due to excretion of succinyl acetone in the urine. Due to excretion of succinyl acetone, tyrosinemia type 1, hepatorenal disease, hereditary hepatorenal condition, tyrosinemia type 1. These are related with the basic metabolism of phenylalanine and tyrosine, in which they will give you glucose and fat. The another important disorder which is related with phenylalanine and tyrosine metabolism with the formation of melanin in the melanin synthesis pathway is the albinism. The disorder is albinism. So we will discuss the albinism. There is generalized hypopigmentation all over the body, and with this the eyesight of the patient is completely normal in the albinism. eyesight, vision is completely normal. So albinism we will discuss. Apart from this inborn error, there are two important disorders which are related with the phenylalanine and tyrosine metabolism. These are, first one is the pheochromocytoma. This is not inborn error, that is pheochromocytoma. This is important disease, and again the Parkinson's disease. These are the important disorders related with phenylalanine and tyrosine metabolism. Now coming to the phenylalanine and tyrosine proper, now see phenylalanine. First, just try to recollect the things about the phenylalanine and tyrosine. Phenylalanine and tyrosine, these are the aromatic amino acids. Aromatic amino acid. What it? See, phenylalanine. Alanine. Okay, to alanine there is benzene ring attached, that is phenylalanine. This is very easy to remember. If you remember the general structure, there is no need to remember the structure. Just see amino group. This is carbon carboxyl group. Amino group carboxyl group. This is H here. This is general structure. In glycine there is H. In alanine there is CH2. In alanine there is CH3. And to this alanine there is attached benzene ring. If benzene ring is attached, it is known as phenylalanine. And if phenol ring is attached, see, if phenol is attached, it is tyrosine. Tyrosine, another name of tyrosine is para-hydroxy phenylalanine. Tyrosine means para-hydroxy phenylalanine. It is produced by the hydroxylation of phenylalanine. Now, these are the aromatic amino acid, non-polar. They belongs to non-polar. Then in metabolic classification, these both phenylalanine and tyrosine, they are included in both glucogenic and ketogenic amino acid. They will give you glucose, they will give you fat, and this phenylalanine is essential amino acid. Essential 10 amino acid, phenylalanine is there, so this is essential. This is non-essential because phenylalanine is converted into tyrosine in our body. So this is phenylalanine and tyrosine introduction part. Now we'll discuss the metabolic pathway, and along with the pathway we'll discuss the disorder, so that we can remember the names of the enzyme, we can remember the name of the accumulated product for that particular disease. Now coming to the pathway, so see the first reaction, phenylalanine is converted into tyrosine. This is important reaction. Phenylalanine is converted into tyrosine. What is another name of tyrosine? It is para-hydroxy phenylalanine. Para-hydroxy phenylalanine. This is another name. Hydroxyl group at the para position of phenylalanine. This reaction, it is catalyzed by phenylalanine hydroxylase. Phenylalanine hydroxylase, monooxygenase. This is monooxygenase. Of two atoms of oxygen, one is incorporated at the para position of phenylalanine and another is reduced to form water molecule. So here O2, it is converted into water molecule. One atom of oxygen is incorporated at the para position while another is reduced to form water molecule. This reaction is tetrahydrobiopterin dependent. PH4, tetrahydrobiopterin dependent. Tetrahydrobiopterin. Okay. In this reaction, tetrahydrobiopterin is converted into dihydrobiopterin and again regenerated with the help of NADPH dependent dihydrobiopterin reductase. NADPH dependent dihydrobiopterin reductase. Dihydrobiopterin reductase. Okay. So this is the important enzyme phenylalanine hydroxylase, which causes hydroxylation of phenylalanine and converts phenylalanine into tyrosine. Now blockade in this reaction. Block in this reaction, it causes classical phenylketonuria. This is classical phenylketonuria. Phenylketonuria. Now what are the symptoms of phenylketonuria and what are the reasons behind the symptoms of phenylketonuria? What are the symptoms? Just try to recollect the symptom first. Mousy odor of urine. This is a characteristic feature. Mousy odor of urine. And the mousy odor of urine, it is because of excretion of phenylacetate in the urine. Second important feature is CNS manifestation, that is mental retardation. Mentally retarded child. Mental retardation is because of accumulated phenylalanine level, and phenylalanine, it is diverted for other product. Phenylalanine is converted into, this is converted into phenylpyruvate. Because this reaction is blocked, so it is converted into phenylpyruvate. It is converted into phenylacetate, phenylacetylglutamine, by the conjugation reaction. Phenylacetate is converted into phenylacetylglutamine. This phenylpyruvate is converted into phenylactate. Okay. So mousy odor of urine is because of this phenylacetate. The keto acid, phenylpyruvate is the keto acid. That's why the name is given phenylketonuria. This is a keto acid. That's why the name is given phenylketonuria. The mousy odor is due to phenylacetate excretion in the urine. Because of accumulation of phenylalanine and blockade in this reaction, there is no synthesis of neurotransmitters dopamine, norepinephrine, epinephrine. Dopamine, norepinephrine, epinephrine, they are not produced. Again, it hampers the tryptophan metabolism. That's why mental retardation is there. There is decreased serotonin production. So these are the two important reasons behind the mental retardation in patient with PKU, phenylketonuria. Then hypopigmentation is there. Hypopigmentation. Generalized hypopigmentation or localized patches in the form of the patches is there. This is because accumulated phenylalanine that inhibit the tyrosinase. So tyrosinase is the important and only enzyme involved in the melanin synthesis. What are the different compounds given by this tyrosine? This tyrosine will give you, see, phenylalanine is converted into tyrosine. Phenylalanine is converted into tyrosine, and this tyrosine will give you, it is glucogenic as well as ketogenic amino. So it will give you glucose, it will give you fat. As these are the amino acids, they will incorporated into a proteins. They will incorporated during the protein synthesis. So they will give you proteins. Now, in their metabolism or the specialized product, this tyrosine will give you melanin in skin, hair and eyes. It will give you melanin. It will give you dopamine, neurotransmitter, dopamine. It will give you norepinephrine, epinephrine. These are the catecholamines. And it will give you T3 and T4 in the thyroid gland, thyroid hormones. These are the specialized product given by the phenylalanine and tyrosine. Now, the only enzyme which is involved in the melanin synthesis is tyrosinase. Here, only tyrosinase is involved. Tyrosinase in this pathway. If there is defective tyrosinase, because increased phenylalanine inhibit the tyrosinase activity, that causes the defective melanin synthesis, that causes hypopigmentation in phenylketonuria. Now see, so these are the three important features in the phenylketonuria: mousy odor of urine, mental retardation, hypopigmented patches all over the body. What are the laboratory tests done for the detection of phenylalanine in urine, or the detection or the diagnosis of phenylketonuria? The most important screening test. Now, at most of the institute, it is replaced by the tandem mass spectrometry and the chromatography, GCMS, gas chromatography mass spectrometry. It is replaced. So confirmatory diagnosis is by the tandem mass spectrometry or the GCMS, gas chromatography mass spectrometry. But the screening test was earlier used. The screening name of the test is Guthrie test. Lab diagnosis is by Guthrie test. In the Guthrie test, this is the bacterial inhibition assay of Bacillus subtilis. Bacillus subtilis assay. बैक्टीरियल इनिबिशन एसे ऑफ बैसिलस सबटिलिस. इसमें क्या करते गुथरी टेस्ट में? हिलप्रिक सैंपल इज कलेक्टेड. हिलप्रिक सैंपल लेते, उसको फिल्टर पेपर पे ब्लॉट करते, है ना. न्यूबॉर्न जो है, न्यूबॉर्न के हिल पे प्रिक करते, उसके बाद वो पूरा फिल्टर पेपर पे ब्लॉट करते, फिल्टर पेपर पे. फिर फिल्टर पेपर को ड्राई होने देते उसको. फिर फिल्टर पेपर ड्राई होने के बाद उसको पंच आउट करते, सर्कुलर डिस्क शेप में उसको पंच आउट करते. पंच आउट करने के बाद एक अगार प्लेट पे बैसिलस सबटिलिस का स्प्रेड करते. बैसिलस सबटिलिस ग्रो होना चाहिए, इस हिसाब से उसको स्प्रेड करते, और वो अगार में एक इनिबिटर रहता जो बैसिलस सबटिलिस की ग्रोथ को इनिबिट करता. ठीक है. नाउ दिस पंच्ड फिल्टर पेपर डिस्क that containing the blood, this disc, they are kept on the agar plate. जो भी डिस्क हमने पंच आउट की है वो अगार प्लेट पे रख देते. नाउ बैसिलस सबटिलिस विल ग्रो बाय टेकिंग द फेनिलैलानिन फ्रॉम द ब्लड. जो फिल्टर पेपर डिस्क में जो ब्लड है वो यूज करके बैसिलस सबटिलिस ग्रो होगा, और उसका कॉलोनी का जो डायमीटर रहेगा दैट करस्पॉन्ड्स विद द कॉन्सेंट्रेशन ऑफ फेनिलैलानिन इन द ब्लड. दिस इज ए बैक्टीरियल इनिबिशन एसे ऑफ बैसिलस सबटिलिस. Bacterial inhibition assay of Bacillus subtilis, Guthrie test, screening test for the diagnosis of phenylketonuria. Now the various tests are available. So GCMS, gas chromatography, mass spectrometry, or the tandem mass spectrometry. These are the laboratory tests which are done for the diagnosis or confirmation of the phenylketonuria. Now the treatment, what treatment is given in the patient with phenylketonuria? Treatment. Which amino acid should be restricted in the diet and which should be given? अभी तक फेनिलैलानिन इंपॉर्टेंट था, है ना. अभी तक हम फेनिलैलानिन इसेंशियल बोलते थे, इसेंशियल अमाइनो एसिड. मैट विल फ्लाई, MATTVILPHLY, मैट विल फ्लाई, 10 अमाइनो एसिड से. 10 अमाइनो एसिड में फेनिलैलानिन भी आता था. अभी तक हम बोलते थे कि फेनिलैलानिन इसेंशियल है, लेकिन अभी तो फेनिलैलानिन जमा हो रहा वहां पे, है ना. सो फेनिलैलानिन शुड बी रिस्ट्रिक्टेड इन द डाइट. टायरोसिन का क्या करना है? टायरोसिन देना है कि नहीं देना है? एज दिस पाथवे इज ब्लॉक्ड, there is deficiency of tyrosine. Now this amino acid becomes essential and tyrosine should be given in the treatment. So low phenylalanine diet, tyrosine should be given in the treatment. Tyrosine should be supplied. Now this amino acid becomes essential amino acid in absence of phenylalanine. Restrict the diet with phenylalanine means जो भी एनिमल प्रोडक्ट्स है, जो भी नॉन वेजिटेरियन डाइट्स है, वो रिड्यूस करो और टायरोसिन का कंटेंट बढ़ाओ. देर आर कॉमर्शियल प्रिपरेशन विच आर phenylalanine free or low in phenylalanine content, so that are available in the market. So low phenylalanine and tyrosine should be given in the treatment, and symptomatic treatment is given. So this is classical phenylketonuria or phenylketonuria. Phenylketonuria type 2, it is due to type 2 and type 3. It is due to defect in the dihydrobiopterin reductase. Defect in the dihydrobiopterin reductase. And 4 and 5, these are related with the tetrahydrobiopterin biosynthesis. 4 and type 5 phenylketonuria. This is type 1. Classical phenylketonuria is the type 1 phenylketonuria. Type 2 and type 3 due to dihydrobiopterin reductase defect, and 4 and 5 it is due to tetrahydrobiopterin biosynthesis. Now see, phenylketonuria type 1, this is classical phenylketonuria due to defect in phenylalanine hydroxylase. Type 2 and type 3, these are due to defect in dihydrobiopterin reductase. And 4 and 5, these are due to tetrahydrobiopterin biosynthesis, related with tetrahydrobiopterin biosynthesis. Got it? Now, what are the important reactions that are tetrahydrobiopterin dependent? Now, remember this reaction BH4. BH4. BH4 मतलब boys hostel 4 नहीं, BH4 means tetrahydrobiopterin. So, tetrahydrobiopterin dependent reactions include, first one is the phenylalanine hydroxylase, then tryptophan hydroxylase, tyrosine hydroxylase, and nitric oxide synthase. These are the 4 important enzymes that requires tetrahydrobiopterin as a cofactor, or coenzyme. Got it? Now, in the symptoms, you may get a blue color eyes. अभी ऐसा मत बोलो कि जैसे ऐश्वर्या राय की ब्लू आईज है, हम बोलते है ना, तो इसका मतलब उसको फिनाइल कीटोन यूरिया है, ये हो गया क्या? द मोस्ट कॉमन कॉज ऑफ blue eyes is congenital. Okay, but sometimes you may get blue eyes, fair hairs, hypopigmented patches all over the body in patient with phenylketonuria. Remember this. Okay, so what are the symptoms of phenylketonuria? Mental retardation. Okay, then hypopigmented patches over the body and mousy odor of urine. The screening test is Guthrie test. Also, ferric chloride test is also done for the screening of phenylketonuria. It is positive for phenylpyruvate, keto acid, phenylpyruvate. Which test? Ferric chloride test. And the confirmation is by the tandem mass spectrometry or gas chromatography mass spectrometry, GCMS. Treatment: low phenylalanine diet, tyrosine should be rich in the diet. Got it? And symptomatic treatment: behavioral therapy, learning therapies, various therapies, occupational therapy, physiotherapy, they are needed. Okay, and these are the 4 tetrahydrobiopterin dependent reaction: phenylalanine hydroxylase, tryptophan hydroxylase, tyrosine hydroxylase, and nitric oxide synthase. These are the types of phenylketonuria. Okay, also this can be diagnosed by assessing the plasma level of phenylalanine by the chromatography. Got it? Now see the pathway now. So this is tyrosine. The another name of tyrosine is para-hydroxyphenylalanine. Now, any amino acid in their metabolism first undergoes transamination process. In their metabolism, first reaction is transamination. So here tyrosine transaminase, with the help of tyrosine transaminase, para-hydroxyphenylalanine means tyrosine. Para-hydroxyphenylalanine is converted into para-hydroxyphenylpyruvate. Amino acid is converted into keto acid. Amino acid is converted into keto acid. In this reaction, there is alpha ketoglutarate, which is converted into glutamate. So glutamate is an acceptor of amino group. All amino group from amino acid, different amino acid, they are channeled or funneled into glutamate, and glutamate undergoes oxidative deamination to liberate the ammonia, which is detoxified by the urea cycle. Remember this. So glutamate, tyrosine is converted into glutamate. So tyrosine will give para-hydroxyphenylpyruvate. And here enzyme name is tyrosine transaminase. Tyrosine transaminase. So as there are 2 T's, tyrosine transaminase. Remember this. This is tyrosinemia type 2. The disorder is tyrosinemia type 2. How to remember this? Tyrosinemia type 2. 2 T's are there. Okay. First, second. So this is tyrosinemia type 2. This shows oculocutaneous manifestations. Oculocutaneous manifestation. Another name of tyrosinemia type 2 is Rischner-Hanhart syndrome. There is oculocutaneous manifestation. Ocular lesions means eye lesion and dermatitis, skin lesion. Oculocutaneous manifestations in this disorder. Oculocutaneous manifestations means eye lesions and dermatitis. Also known as Reisner-Hanhart syndrome, tyrosinemia type 2, due to the defect in tyrosine transaminase enzyme. Defect in tyrosine transaminase enzyme. Okay. So tyrosinemia type 2. Now this parahydroxyphenylpyruvate, it is converted into homogentisic acid. Got it? Now the enzyme here is parahydroxyphenylpyruvate, hydroxylase. This is enzyme parahydroxyphenylpyruvate hydroxylase or dioxygenase. It is also known as dioxygenase. Dioxygenase means incorporates both molecules or both atoms of oxygen into the substrate. Parahydroxyphenylpyruvate hydroxylase. This reaction, hydroxylation reaction, requires vitamin C. So here parahydroxyphenylpyruvate is converted into homogentisic acid. If there is defect in this enzyme, parahydroxyphenylpyruvate hydroxylase, it causes the disorder that is known as neonatal tyrosinemia. Or tyrosinemia type 3. This is type 3 tyrosinemia. This was type 2. This is type 3 tyrosinemia, neonatal tyrosinemia, due to defect in parahydroxyphenylpyruvate hydroxylase. This is dioxygenase. Dioxygenase incorporates both atom of oxygen into the substrate. There is formation of homogentisic acid. Now homogentisate oxidase is the enzyme. Again, it is dioxygenase. Homogentisate oxidase, it is converted into 4-methyl acetoacetate. The here enzyme is homogentisate oxidase. Important disorder, homogentisate oxidase. Deficiency causes important disorder. The name of the disorder is 𝐴𝐾aptonuria. If there is defect in homogentisate oxidase, it causes 𝐴𝐾aptonuria. Now what is 𝐴𝐾aptonuria? We'll discuss this, just wait. It is included in the Garrod's tetrad. 𝐴𝐾aptonuria. It is included in the Garrod's tetrad. Garrod's tetrad includes 4 disorders. One is 𝐴𝐾aptonuria, then essential pentosuria, cystinuria, and albinism. 𝐴𝐾aptonuria, essential pentosuria, albinism, and one more. See, Garrod's tetrad. It is included in the Garrod's tetrad, that is essential pentosuria, then albinism, cystinuria. Cystinuria is the most common inborn error of metabolism. Frequency is 1 in 7,000. There is excretion of cola in urine, COLA. Cysteine, ornithine, lysine, arginine in the urine due to defective reabsorption from the renal tubule. Cystinuria. Okay. PACA, that is 𝐴𝐾aptonuria. So these are included in the Garrod's tetrad. Okay. Now 𝐴𝐾aptonuria. What is 𝐴𝐾aptonuria? This is autosomal recessive disorder due to the defect in the enzyme homogentisate oxidase. The another name of homogentisate oxidase, just remember this, homogentisate dioxygenase. Dioxygenase, this is dioxygenase. It requires iron. It requires iron. Homogentisate dioxygenase. Or another name is, this is important for the INICET exam. So homogentisate, this is, see, para-hydroxyphenylpyruvate. Okay. This is para-hydroxyphenyl acetate. So here enzyme name is para-hydroxyphenyl acetate dioxygenase. The name of the enzyme, another name is the para-hydroxyphenyl acetate dioxygenase, the another name of homogentisate oxidase. Now what occurs or what happens in the alkaptone urea? Just wait. This is autosomal recessive disorder, asymptomatic, most of the time asymptomatic, often goes unnoticed. This is asymptomatic and often goes unnoticed. Just wait. So this one is autosomal recessive disorder and often goes unnoticed because what is the principal manifestation in alkaptonuria? क्या manifestation है alkaptonuria में? Dark urine on exposure to air. Okay. Now it is often goes unnoticed because it takes some time. It takes some time. Now in alkaptonuria, because of defect in this enzyme, so this is homogentisic acid. So homogentisic acid is converted into 4-mallyl acetoacetate. Acetoacetate. Now there is block in this reaction. So this homogentisic acid, it is converted into, it goes to alternate pathway and it is converted into benzoquinone acetate. Benzoquinone acetate. And this benzoquinone acetate, by the polymerization, it is converted into alkaptone. It is converted into alkaptone. This is black pigment. Alkaptone. Black pigment. Alkaptone on polymerization. So homogentisic acid, by the polyphenol oxidase, it is converted into benzoquinone acetate. Benzoquinone acetate on polymerization gives alkaptone, and alkaptone deposited in the cartilage, tendon, bone, joints. It is deposited in the tendon, sclera, bones, joints, and it causes arthritis. Arthritis also gives the black discoloration of cartilage of the nose, ear, legs. It gives the black discoloration. So this is the reason for the black color pigment formation, because benzoquinone acetate on polymerization give alkaptone. This is the alternate route of homogentisic acid. Urine turns dark on standing, or alkalinization of urine, it becomes dark or brown color, black color or brown color urine because of homogentisate oxidase. This is autosomal recessive disorder, often goes unnoticed. You may notice a black staining diaper. Okay, autosomal recessive, urine turns dark on standing, on exposure to air, urine turns dark. So this is classical feature. Then there is tendency to develop arthritis in the adulthood. Arthritis. So there is black deposition in tendons, sclera, bones, joints, that causes the arthritis. Also there is deposition into the intervertebral disc, that causes the arthritis. So this is important disorder, alkaptonuria. It is included in the Garrod's tetrad. Now what is the treatment? The symptomatic treatment is given for the arthritis. Treatment: symptomatic treatment is given. Now new drug is there. Symptomatic treatment is given, and now new drug is there which is given in the treatment. The name of the drug is nitisinone. Nitisinone. This is a new drug which is given in the treatment in patient with alkaptonuria to reduce the symptoms. What is the mechanism of nitisinone? See, this is pathway. This is pathway. Nitisinone inhibits this para-hydroxy, inhibit this nitisinone. This drug inhibit it. This drug nitisinone inhibit the para-hydroxy phenylpyruvate hydroxylase and decreases the production of homogentic acid. So it relieves or it reduces the symptoms of alkaptonuria. Inhibit the para-hydroxy phenylpyruvate hydroxylase and decreases the production of homogentic acid. So accumulated homogentic acid will be less. So this is drug which is given in the treatment of alkaptonuria, nitisinone. Okay. Now laboratory test for the diagnosis. Laboratory test. The ferric chloride test may be positive. Ferric chloride test. Then reducing means Benedict test can be positive because of homogentic acid. Because of reducing property, homogentic acid will give the Benedict test also positive. Positive Benedict test. Okay, but confirmatory diagnosis is by the amino acid assay, that can be by the chromatography or by the tandem mass spectrometry, TMS, GCMS or chromatography. This is confirmatory test for the diagnosis of the alkaptonuria. So asymptomatic in the childhood, at the age in the 3rd to 4th decade of life, there is tendency to develop arthritis. Symptomatic treatment for arthritis is given, and this is a newer drug which decreases the para-hydroxy phenylpyruvate hydroxylase level and decreases the homogentic acid production. So alkaptonuria, it is important disorder in the inborn error of phenylalanine and tyrosine. Now see the pathway again. Now 4-mallyl acetoacetate, it is converted into fumaryl acetoacetate. 4-fumaryl acetoacetate, and fumaryl acetoacetate, it is hydrolyzed by hydrolase, fumaryl acetoacetate hydrolase, and it is converted into fumarate. It is converted into fumarate and acetoacetate. Fumarate will give you glucose and acetoacetate will give you fat. This will give you fat. This will give you glucose. Through the TCA cycle, it will give you glucose. TCA cycle and gluconeogenesis, it will give you glucose. So fumarate and acetoacetate. Here enzyme is fumaryl acetoacetate. Fumaryl acetoacetate hydrolase. Fumaryl acetoacetate hydrolase. That will cleave the fumaryl acetoacetate into fumarate and acetoacetate. So in this way, this phenylalanine and tyrosine, they are both glucogenic and ketogenic. Because they give glucose, they will give you fat. This is isomerase here. So because of deficiency of fumaryl acetoacetate hydrolase, the defect is known as, the defect here, if there is block here, the defect is known as tyrosinemia type 1. The defect is known as tyrosinemia type 1. This is known as hereditary tyrosinemia. Hereditary tyrosinemia. This is known as hereditary tyrosinemia, hepatorenal tyrosinemia. Hereditary tyrosinemia, hepatorenal tyrosinemia. Because of defect in the enzyme fumaryl acetoacetate hydrolase. This enzyme is defective, that affect the liver, that affect the kidneys, and there is cabbage like odor of the urine and body because of excretion of succinyl acetone. Because of excretion of succinyl acetone, this may be converted into succinyl acetone, and that gives the boiled cabbage like odor of the urine in the tyrosinemia type 1. Boiled cabbage like odor. Also in this disorder, nitisinone can be given. Nitisinone is the drug which decreases the level of this homogentic acid, so further products will not form. So nitisinone is also given in the tyrosinemia type 1. Now this, remember this disorder. See, you know phenylalanine hydroxylase, phenylketonuria. Two T's are there, tyrosine transaminase, tyrosinemia type 2. Two T's are there, tyrosinemia type 2. This is transamination reaction. Then this is hydroxylation reaction. This is vitamin C dependent reaction. Just wait. This is vitamin C dependent reaction, hydroxylation reaction. Okay, para-hydroxyphenyl pyruvate hydroxylase. It causes neonatal tyrosinemia or tyrosinemia type 3. Then homogenticate oxidase defect, it causes 𝐴𝐾 autosomal recessive disorder included in the Garrot's tetrad, asymptomatic in the childhood. There is tendency to develop arthritis because of deposition of 𝐴𝐾 in the joints, tendons, basically in the joints, that causes arthritis. Also there is black discoloration of ear cartilage, nose cartilage, black coloration of legs. So black discoloration is due to 𝐴𝐾, euchronosis. This condition is known as euchronosis. Okay. Then this 4-mallyl acetoacetate is converted into or isomerized into fumaryl acetoacetate. Fumaryl acetoacetate is breakdown into fumarate and acetoacetate. Fumarate via the TCA cycle and gluconeogenesis will give you glucose, and acetoacetate will be converted into fat. The defect in fumaryl acetoacetate hydrolase causes the tyrosinemia type 1. It is hereditary tyrosinemia, also known as hepatorenal tyrosinemia. It is hepatorenal tyrosinemia. There is cabbage-like, boiled cabbage-like odor in the tyrosinemia type 1. Now the classical feature, mousy odor in the phenylketonuria. Urine turn dark on standing. It is in the 𝐴𝐾. Boiled cabbage-like odor. It is tyrosinemia type 1. It is tyrosinemia type 1. What are the reactions in which the this tetrahydrobiopterin is the coenzyme? Remember the amino acids: phenylalanine hydroxylase, tyrosine hydroxylase, tryptophan hydroxylase, and nitric oxide synthase. Nitric oxide synthase. These are the 4 important enzymes which require tetrahydrobiopterin as a cofactor. What is Garrot's tetrad? Garrot's tetrad means the 4 disorders that are included in the Garrot's. That is essential pentosuria. There is excretion of xylulose in the urine. Albinism, which is generalized hypopigmentation due to defect in the tyrosinase activity. Cystinuria, it is defective renal tubular reabsorption of the 4 amino acid, that is CoLA. Cysteine, ornithine, lysine, arginine. Renal tubular reabsorption, cystinuria, 1 in 7,000 frequency. 𝐴𝐾, it is autosomal recessive disorder due to defect in homogenticate oxidate. So this is Garrot's tetrad. What are the symptoms of tyrosinemia type 2? It is also known as Richner-Hanhart syndrome. There is oculocutaneous manifestation that can be include palmar hyperkeratosis, palmoplantar hyperkeratosis, also eye lesions, also dermatitis. Okay, so this is tyrosinemia type 2. Then neonatal tyrosinemia, then the tyrosinemia type 1, boiled cabbage like odor. Okay, so this is the mechanism behind the euchronosis, that is deposition of 𝐴𝐾. Homogenticate, it is converted into benzoquinone acetate by the polyphenol oxidase, and this benzoquinone acetate on polymerization gives the 𝐴𝐾 black pigment, which is deposited in the 𝐴𝐾. So this is new drug nitisinone, which is given in the treatment of 𝐴𝐾 and the disorders. Okay, now the albinism. Generalized hypopigmentation. अल्बिनिज्म, जनरलाइज्ड हाइपोपिगमेंटेशन. आप सबने अल्बिनिज्म का पेशेंट देखा ही होगा. जनरलाइज्ड हाइपोपिगमेंटेशन along with fair hair, fair eyes, then the ocular manifestation will be there, but visual acuity or the eyesight will be normal in the albinism patient. Okay, this is due to the defect in the enzyme, that is tyrosinase. Defect in the tyrosinase. So defect in the tyrosinase. How melanin synthesis occur? This is a pathway for melanin synthesis in which a tyrosine, it is converted into DOPA, that is dihydroxyphenylalanine, DOPA. Tyrosine is converted into DOPA by the tyrosinase. DOPA is converted into dopaquinone, which is converted into leukodopachrome, and that will give you the melanin. Red melanin, black melanin, that will give you melanin. So melanin synthesis, two important means only one enzyme is there, that is tyrosinase, and if there is defect in the tyrosinase activity, it causes the albinism. Means decreased melanin synthesis or defective melanin synthesis is related with the albinism, tyrosinase defect. And what are the main clinical features of albinism? It includes hypopigmentation, generalized. If it is generalized hypopigmentation, then fair hair, eyes, okay, then photophobia, intolerance to the sunlight, photophobia will be there. So these are the manifestations of albinism. There is the clinical diagnosis. You can obviously diagnose the patient with the albinism. There is no need of any laboratory test for the diagnosis of albinism. The localized hypopigmentation, it is known as vitiligo or leukoderma. Vitiligo, it is known as vitiligo, localized hypopigmentation. So there is defective tyrosinase activity. Apart from these disorders, there are two important disorders related with phenylalanine and tyrosine, but these are not inborn error. First one is the pheochromocytoma. This is involving the adrenal gland, pheochromocytoma, and it is related with catecholamine synthesis, epinephrine, norepinephrine synthesis. And second important disorder related with this amino acid, phenylalanine and tyrosine, is the Parkinson's disease. These are the two important disorders related with phenylalanine and tyrosine specialized product. What is pheochromocytoma? In the pheochromocytoma, there is, what are the symptoms? The symptoms are palpitation, headache, and raised blood pressure. And there is increased excretion of vanillyl mandelic acid in the urine. Excretion of VMA in the urine, vanillyl mandelic acid in the urine. There is increased level of catecholamines. Catecholamines means epinephrine, norepinephrine in blood. Increased level of catecholamines in the blood, increased excretion of VMA in urine. So these are the features of pheochromocytoma: palpitation, headache, raised blood pressure. Here, how epinephrine and norepinephrine they are formed. See, this is the simple pathway in which tyrosine is hydroxylated to form the DOPA, dihydroxyphenylalanine, with the help of tyrosine hydroxylase. This enzyme is tyrosine hydroxylase. This is tetrahydrobiopterin dependent reaction. It is converted into dihydroxyphenylalanine, that is DOPA. DOPA is converted into dopamine. DOPA decarboxylase, it is converted into dopamine. Decarboxylation reaction. Pyridoxal phosphate dependent decarboxylation reaction. Remember, pyridoxal phosphate is required for the decarboxylation reaction, PLP. It is converted into dopamine. This dopamine hydroxylase, it is converted into, by the dopamine hydroxylase, it is converted into norepinephrine. Epinephrine, and norepinephrine is converted into epinephrine, means adrenaline. This is tumor of adrenal gland. There is increased catecholamine synthesis, norepinephrine and epinephrine. So elevated level of norepinephrine and epinephrine in the blood, and this norepinephrine, epinephrine, dopamine, they are metabolized by the, with the help of two important enzymes, that is methyl. Wait, this COMT, catechol O methyl transferase. They are degraded by the catechol O methyl transferase into normetanephrine and metanephrine, and again acted upon by the monoamine oxidase, and it is converted into vanillyl mandelic acid. So excretion of VMA in the urine is a diagnostic test for the pheochromocytoma with this classical feature: palpitation, headache, raised blood pressure. Palpitation, headache and raised blood pressure, these are the clinical features. Increased catecholamines in the blood, increased excretion of VMA, vanillyl mandelic acid, in the urine. It is related with the catecholamine synthesis of dopamine, norepinephrine, epinephrine. Okay. So this dopa decarboxylase, it is pyridoxal phosphate dependent reaction, dopamine hydroxylase, vitamin C dependent reaction, and norepinephrine epinephrine conversion. This is methyl transferase reaction, in which methyl group is donated by the S-adenosyl methionine. S-adenosyl methionine. Yes, adenosyl methionine. This is hydroxylase. Remember, norepinephrine, no methyl group, norepinephrine, no methyl group, epinephrine, there is methyl group. Okay. Now Parkinson's disease. What do you mean by Parkinson's disease? There is decreased dopamine. This is because of decreased dopamine, because of age-related disorder, because of age-related degeneration of the basal ganglion. Certain areas of the brain, that includes the basal ganglion. So decreased dopamine level because of degeneration. Near about the 1% of population, degeneration of certain areas of the brain, particularly basal ganglion, that affect the 1% of population above the age of 60 years. 1%, मतलब 100 में से एक. Parkinson's disease. Typical features are there: mask-like face, rigidity, tremors, involuntary movements. These are the features of Parkinson's disease. Mask-like face, rigidity, tremors, involuntary movements are there. Okay, gait disturbance is there in the Parkinson's disease. Decreased dopamine in certain areas of the brain. Okay, so dopamine decrease. There is decreased dopamine level. And what is given in the treatment? This is connected with pharmacology. So L-dopa is given in the treatment. L-dopa, levodopa is given in the treatment. Levodopa crosses the blood-brain barrier, enters into the brain, where it is converted into dopamine. So it increases the level of dopamine in the brain. Along with the levodopa, carbidopa is given. What is role of carbidopa? Carbidopa prevents the peripheral conversion of L-dopa into dopamine. बाकी सब बॉडी में पर इसका conversion prevent करता है carbidopa. So it prevents the side effect of the L-dopa. Side effects of dopamine in it prevents. Okay, so L-dopa along with the carbidopa is given in the treatment. L-dopa crosses the blood-brain barrier, enters the brain, where it is converted into dopamine and elevate the level of dopamine, decreases the symptoms of Parkinson's disease. Carbidopa prevent the peripheral conversion. Apart from the brain, it prevent the carbidopa does not crosses the blood-brain barrier. So in the other part of the body, carbidopa prevents the conversion of L-dopa into dopamine and prevents the side effect of this L-dopa. So this is given in the treatment. Parkinson's disease. Mask-like face, involuntary movement, tremors, rigidity. That is typically cogwheel rigidity. That is known as cogwheel rigidity. Okay. So this is Parkinson's disease. So what we learned today, see, summary, what we learned today, phenylketonuria, it is common, important, inborn error of phenylalanine and tyrosine metabolism due to defect in phenylalanine hydroxylase. The classical feature is mousy odor of urine. Mousy odor of urine. Then the second important disorder is alkaptonuria. Alkaptonuria. It is due to homogentisate oxidase defect, asymptomatic during the childhood. Later on, there is tendency to develop arthritis due to ochronosis, due to deposition of alkaptone pigment. Here, the characteristic feature is urine turns dark, urine on standing, black urine on standing. Black urine on standing. This is a feature, black staining of diaper in alkaptonuria. Then tyrosinemia. There are three types of tyrosinemia. The important one is tyrosinemia type 2, that is tyrosine transaminase defect. It is also known as Richner-Hanhart syndrome. Oculocutaneous manifestations are there. Palmoplantar hyperkeratosis is there, is a feature of tyrosinemia type 2. Then tyrosinemia type 1, there is boiled cabbage-like odor. Tyrosinemia type 1, there is boiled cabbage-like odor in tyrosinemia type 1. Hepatorenal disease, tyrosinemia type 1 due to defect in fumarate acetoacetate hydrolase. Okay. Then albinism. What is albinism? Albinism, it is tyrosinase defect. There is defect in the tyrosinase. Generalized hypopigmentation is there in the albinism. Got it? So these are the important disorders related with the phenylalanine and tyrosine metabolism. So this is all about the phenylalanine and tyrosine metabolism. Thank you. Just wait. Thank you.